Hi dear friends, welcome to my channel Mitul's Biolog. Hope you all are fine and safe. Today we are with a new topic, that is reproduction in flowering plants. So welcome to the section. Here this is the first part of this topic. Here I will mention about different parts of flower and reproductive part of flower in detail. So flowers are the modified shoot of a plant for reproduction. We want to study what are the different parts of a typical flower. Let us see one by one. A flower is having four main parts. One is calyx, then another one is corolla, third one is gynoecium, and the fourth part is anther. Now let us see one by one in detail. Here there is a flow chart given showing the complete part of a typical flower. First of all, the outermost part of the flower, which is arranged in a whorl, that is in a circular form, is called calyx. So calyx is the outermost part of a flower. They are made up of sepals. The green colored portion is called a sepal. So one or more sepals can be seen, and sometimes the sepals will be fused together, or sometimes it will be not fused, it will be free. So that is the first part of a flower, and their function is to protect the bud. That is when bud condition, the protection is provided by the calyx. Okay, now the next inner part, second part or second whorl, inner whorl, is called as corolla. Corolla is made up of the colored attractive petals. These petals are the colored attractive portion of a flower, and their function is to carry out pollination. Obviously we know this part will attract the insects for pollination. Now these two parts, the corolla and calyx, they are called as the sterile part of a flower, and so because of that they are called non-essential whorls of a flower. Clear? The third part of a flower is anther. Anther is the male reproductive part, and they are made up of individual unit called stamen. Numerous stamens can be seen in a flower. Stamen is having three main parts: anther, connective, and filament. The next important part of a flower is gynoecium. Gynoecium is the female reproductive part, and they are made up of individual units called carpels, or it is called as pistil. Carpel is having three main parts: stigma, style, and ovary. These two parts, that is anther and gynoecium, since they undergo sexual reproduction, they are called as the essential whorl of a typical flower because they are the fertile part of a flower. So these are the parts of a typical flower. Here this is the diagrammatic representation of a typical flower. All parts are mentioned here: corolla, calyx, anther, gynoecium, where it is present, everything. Now next we have to study in detail about the sexual reproductive part of a flower. First we comes to the anther, which is made up of stamens. Stamen is also called as microsporogenesis. Now stamen is having three main parts, already I mentioned. First part is the anther. Anther is a bilobed structure which will be seen at the tip of the stamen, and this portion is connected with a stalk which is called as filament. Anthers with the two lobes are connected by a connective tissue, which is a paragynematous tissue which is seen in between the two lobes of the anther. Here, normally in most of the plants we can see bilobed anther. The bilobed anthers are called as dithecous anther. There are plants with single lobed, and that type of anthers are called monothecous anther. Example is China rose. Over this anther, we can see yellow powder which is called as pollen that undergo pollination. This pollen will produce the male gamete and undergo the sexual reproduction. Now we have to study the structure of anther in detail. Already I mentioned anther is having two lobes, and these two lobes itself, they are four-sided structure. Over the four sides you can see four pollen sacs. Four pollen sacs are arranged in four sides of the anther. Now anther is provided with four whorls. They are called anther whorls around the anther. Let us see in detail. Just focus upon to one part of the anther. The first layer is epidermis. Epidermis is the outermost protective layer. Then comes the second layer, which is shown in the purple color. It's called endothecium. endothecium is the second layer just seen below the epidermis, and this is a hypodermal layer. They help for the breaking of anther, that is called dehiscence of anther when it gets matured for the dispersal of pollen. Then comes the third layer, which is called as middle layer, and it's just shown in blue color. They are of 1 to 3 layer in thickness. And last, the fourth layer, it's called tapetum. Tapetum is a nutritive layer, will be seen around all the pollen sac, or for all 4 pollen sac. Around the pollen sac you can see this tapetum. Then at the center we can see the 2 lobes are connected by a parenchymatous tissue, which is called as connective. They connect the 2 lobes of the anther. So these are the different parts of anther. This is a typical structure of anther. You can just note it down. Now with this we have to study what is the development of anther, how the anther develop and how they produce the male reproductive cells. Here the cells at the 4 corners of anther, that is the archisporal cells, undergo periclinal division. Now what is periclinal division? Periclinal division is a type of cell division in which the cells will be divided parallel to the tissue, and as a result numerous rows of cells will be formed, one above the other. In this way, this archisporal cells divides and they form parietal cells and sporogenous cells. The outer formed cells are called as parietal cells, and the cells which is formed to inside, which is called as sporogenous cells. Likewise, rows of cells will be formed. They are differentiated as outer parietal cells and inner sporogenous cells. The rows of cells which is formed towards outside, that is called as a parietal cells, later transforms to walls of anther, that is the epidermis, endothecium, middle layer and tapetum, and the inner sporogenous cells later transforms into microspore mother cells. They are the sporogenous cells, which is called as microspore mother cells or pollen mother cells. So in this way the anther walls and microspore mother cells are getting differentiated. Clear? What is the function of this microspore mother cell? Microspore mother cell is the particular cell which will produce microspore or pollen. The process of production of microspore or pollen by this microspore mother cell is called as microsporogenesis. Now we have to study how this happens. Each microspore mother cell in the pollen sac undergo meiosis division, and as a result 4 daughter cells will be formed. We know meiosis happens in 2 stages, meiosis 1 and meiosis 2. In meiosis 1, first they divide into 2 daughter cells, it is a dyad form. Then again they undergo meiotic 2 division and they form 4 daughter cells. Here from 1 microspore mother cell, 4 microspores are formed, that is 4 pollen grains are formed, and they are in the form of a tetrad, that is in a 4 embedded structure. So this is a formation of microspore from 1 microspore mother cell. So obviously you can imagine inside the pollen sac numerous microspore mother cells are there. So how many microspores will be formed? Numerous microspores will be formed from a pollen sac. There are 4 pollen sacs in an anther. So just imagine what will be the process. We have to study about pollen grain in detail. Pollen grain or microspore, they are variable in sizes, shapes and ornamentation. That is, the texture will be different for each pollen. The pollen is actually the first cell of male gametophyte. Pollen cause allergies and also many respiratory diseases. Example is Parthenium grass, which cause pollen allergy. Then the pollen tablets are available, which are used by athletes and also given for race horses because that protein enhance the power. The study of pollen is called palynology. Next, let us study the structure of pollen grain. Mature pollen grain has a double-walled structure. The wall of pollen grain is also called as sporoderm. The outer wall is called as exine or exospore. It's a tough-resistant wall composed of sporopolanin. The sporopolanin is actually a resistant polymer, and because of this sporopolanin they are non-degradable, and the exine wall is variously sculptured. The next is intine. Intine is a delicate wall of cellulose which is seen just below the exine. Intine wall is also called as endospore. The next, there is another portion which is called aperture. Apertures are the region of the pollen wall where exine is reduced in thickness, and this allows the shrinking and swelling of the pollen caused by the changes in the moisture content in the atmosphere. Elongated apertures are called germinal furrows, whereas circular apertures are called as germ pores. Okay. Next, let us study how this pollen grain produces the male gametes. The process of production of male gametes by the pollen grain is called microgametogenesis. Now let us see how this happens. Okay, now how is the development of male gametophyte? The nucleus of the pollen grain undergo mitotic division, and as a result they develop two daughter cells. One is the larger daughter cell is called as vegetative cell or tube cell, and the smaller cell is called as generative cell. Here the arrangement is the smaller cell is seen in the larger cell. That is, it's a bipolar structure, a cell within a cell. So that is the arrangement of the two cells in the pollen. This generative cell is seen towards the base of the pollen grain. After this mitotic division, in this two-celled stage condition, this pollen grain undergo pollination. In most of the angiosperm, the pollination is happening in this two-celled stage. So obviously the pollen grain will fall over the stigma, and the pollen grain start to absorb the nutrient from the stigma through the aperture, and at the time the vegetative cell or the tube cell starts to enlarge. Then once it is enlarged, the intine wall is moved out through the germ pore, forming pollen tube. So this is happening next. Once the pollen tube is formed, the vegetative cell nucleus and the generative cell nucleus move into the pollen tube, and this generative cell nucleus undergo the second mitotic division, and as a result they form two male gametes. Okay. In few plants the condition is different. Pollination happens only at this three-cell stage. That is, only after the formation of male gametes pollination happens. But in majority of angiosperm, pollination happens at the early mitotic division, that is in two-cell condition. Here pollen grain with pollen tube, vegetative nucleus and two male gametes, together it is called as male gametophyte. In this picture you can see the male gametophyte. Next, what is pollen viability? Pollen viability is the ability of the pollen to transfer male gamete to the embryo sac, that is to the ovule of the flower. Viability is always related with the environmental temperature and humidity condition, and this viability of pollen changes from species to species, and it is also related with the type of pollination. The pollen viability of rice and wheat plant is about 30 minutes. Okay. Now how we can maintain the pollen viability? The method is by cryopreservation. Cryopreservation is a method of storing pollen in liquid nitrogen under -196 degrees Celsius, and it can be stored for many years in pollen bank. You might have studied or heard about gene banks. Similar way, there are pollen banks for storing pollen. Normally we use this method for crossbreeding programs and also for many research studies. Okay, so in this video I have mentioned all about the male reproductive part and their development. The remaining portion I will upload in the next video about the female reproductive part and how they help for the reproduction, that is their development. Okay, so thank you for watching my video. I hope it was helpful for you. If you liked my video, please like, share and subscribe, and thank you for watching my video once again. Okay, bye, see you.